On the 25th of March, 2026, a medical journal described 5 people who each received a single infusion. No cells were removed from their bodies. No chemotherapy cleared space first. What went in was a vector, an engineered courier carrying one set of instructions for finding their cancer, and their own T cells did the rest. Inside their bodies, they built the receptor and became cancer-hunting CAR-T cells. One manufacturing step moved inside the patient. For a treatment like this, that has never happened before. To be clear about what did not happen, the body invented nothing. Scientists designed the receptor, a facility made and tested the vector. Clinicians delivered it and managed what it did, but one step did move. And if one step can move, it is worth asking precisely, not poetically, what it takes for a living drug to be made and made available. Not, does it work? Can anyone get it? Because today, making this medicine is a journey. It begins with leukapheresis. The patient's blood leaves their body, a machine separates out the white cells, and the rest goes back. Those T cells travel to a facility. They are activated, given the new receptor, and grown into a treatment dose. Then they are checked. Are these the right patient's cells? Did the modification work? Are they potent, pure, clean? Only then, frozen, shipped back. After chemotherapy to make room, are they infused? The National Cancer Institute puts the round trip at about 3 to 5 weeks. A bespoke supply chain for an audience of one. Medicine has been here before. In London in 1928, Alexander Fleming came back to a contaminated culture dish and saw something odd, a ring of dead bacteria around a blob of Penicillium mold. He published the effect a year later, but an effect in a dish is not a medicine. Fleming's stuff was unstable, difficult to isolate, and available only in crude, tiny amounts. The medicine did not exist, and for 10 years, almost nothing happened. In 1939, a team at Oxford took the problem apart. Howard Florey organized the work. Ernst Chain and Edward Abraham went after the chemistry. Norman Heatley invented ways to grow the mold, extract the penicillin, and measure it. They purified enough to run real experiments. In mice, the results were dramatic. They prepared the first doses for people. Fleming had found the effect. Oxford made the effect usable, but usable is not the same as available. Making a little penicillin in a laboratory was an exhausting, hand-built process, and exhaustion is not a supply. On the 12th of February 1941, the Oxford team began treating Albert Alexander, a policeman gravely ill with infection. The treatment worked. His fever fell, his appetite came back, and the reservoir drained away. Every drop of penicillin they had went into him. When it ran low, they recovered what they could from his urine, purified it, and put it back. It was not enough. The supply ran out before the treatment was finished. The infection returned, and Albert Alexander died. Hold this carefully. It is not a horror story, and it is not a legend. It is the exact measurement of a gap. They had proof the drug worked, and no repeatable way to make the amount one man needed. That is the distance between efficacy and availability. So in the summer of 1941, with a war on, Florey and Heatley carried the problem across the Atlantic to Peoria, Illinois, and a Department of Agriculture laboratory that understood industrial fermentation. First, Peoria changed the food. Corn steep liquor, a rich byproduct of corn processing, fed the mold far better, and output jumped. Then it changed the vessel. Instead of growing mold on the surface of hundreds of shallow dishes, teams pursued submerged fermentation, mold growing through deep tanks of liquid. That meant controlling oxygen, mixing, and temperature at scale. A bigger container is not, by itself, a process. And then, in 1943, the famous part. Somewhere in Peoria, at a local market, someone found a moldy cantaloupe. On it grew a Penicillium strain catalogued as NRRL 1951, and it was better suited than Fleming's strain to producing penicillin in those deep tanks. Here is the honest version of the legend. The cantaloupe did not make penicillin. It gave a system that scientists and engineers were already building a much better starting point. Luck delivered a melon. What happened next was not luck. Researchers selected the cantaloupe strain's most productive descendants, then pushed them further with X-rays and ultraviolet light. Engineers solved aeration, cooling, extraction, purification, contamination control, and scale-up. Laboratories, universities, and pharmaceutical companies shared methods. On the 1st of March, 1944, Pfizer opened a commercial plant for large-scale submerged production. by D-Day. Penicillin was available for severe Allied casualties. 3 years after Albert Alexander, the gap he measured had been closed, not by a discovery, but by a supply chain. Lay the whole story out and it has 5 links. Each one made the next possible. Prove the effect works, the clearing, the mice, the first doses. Make it reproducibly, the food, the vessel, the strain, the factories. Verify what was made, Heatley's measurements, every batch checked. Move it without losing it across an ocean into hospitals. Reach the patient, Albert Alexander, then the wounded of D-Day. Prove, make, verify, move, reach. Penicillin crossed all 5. Now come back to the medicine we started with, because it crosses the same 5 links, except the product is alive, and it belongs to one person. A conventional pill can be made in batches. Autologous CAR T cells begin as one patient's living material and only ever return to that patient. Chain of identity is part of the medicine. The cells are collected, activated, modified, expanded, and tested. Identity, viability, purity, potency. Safety before the dose is released. Meanwhile, the patient waits, and the disease does not. Before infusion, patients generally receive chemotherapy to make room, and the clock is 2 clocks. The Institute's 3 to 5 week estimate covers collection to infusion. Its Frederick program describes a narrower 2-week manufacturing and testing segment after material reaches a centralized facility. Those are not interchangeable numbers. So how do you move a factory? There are 3 answers on the table. 1. Share it. In the Frederick program, hospitals ship frozen patient material to a centralized, closed facility, which manufactures, tests, and returns the cells. 1 strong factory shared. 2. Build it closer. India's next CAR-19 was designed through collaboration between Indian researchers and institute colleagues and is manufactured in India, still an autologous. Outside the body therapy, not a bedside process. The factory moves nearer the patients. 3. Move a step inside. Instead of removing, engineering, and returning the cells, infuse a targeted vector and let the body do the last step. That is the 2026 experiment. Note what it changes and what it does not. The vector still has to be made, tested, moved, and administered correctly. Manufacturing did not disappear. So the results and their limits with equal weight. This was a phase 1 single-arm open-label study, 5 heavily pretreated men after a median of 3 previous treatment lines with a median follow-up of 6 months. Its primary purpose was safety and tolerability. 4 of 5 had an objective response. 3 had a stringent, complete response. All 4 evaluable responders were minimal residual disease negative by day 60. Preliminary signals, nothing more, and the harms. All 5 had grade 3 or higher adverse events. 4 developed cytokine release syndrome, 3 at grade 3, 1 at grade 2. 3 had grade 2 infections. 1 had grade 1 neurotoxicity, and 1 patient died from spinal cord compression related to an extramedullary lesion, the paper reports. The trial stopped early in 2025. The paper does not say why. A registry last updated in February 2025 still says recruiting. Neither fact tells us a reason, and we will not invent one. 5 people are not a verdict, not on efficacy, safety, durability, manufacturability, or access. What they show is smaller and still astonishing. 1 boundary of the factory moved. A clearing in 1928 showed an effect. An empty reservoir in 1941 exposed the real problem. A cantaloupe in 1943 improved 1 input. A system, food, vessel, strain, process, factories made penicillin available. The experiment of 2026 moves 1 step of that system inside the patient. Everything after it must still be proved, made, verified, moved, and reached. That is how an effect becomes a medicine. It never was the melon. It was everything around the melon, and that is what makes the next step less impossible. More at lessimpossible.com.